Alright, ladies and gentlemen, well, I hope everyone's doing well here. Today we're going to take a look at classification. So before we begin, take a moment to think about some of the big questions that we're going to be looking at here. We want to make sure that in this chapter we get a sense of how individuals can go about trying to classify organisms based on their evolutionary relationships. In the last unit we looked at mechanisms by which populations can evolve and the ways in which new species are created. And today we're going to look at how do you actually go about classifying all these different species that have created. So you get to look at really important questions like this one here. Is it a snake or a lizard? If you've read the chapter, you already know the answer. So for those that have, kudos. Alright, so let's begin by looking at how things have been done for the last few hundred years. So Carolus Linnaeus was a European botanist who would on occasion take off the powdered wig and head out into the woods and look at different types of plant species that he could classify. His goal was to classify all the different forms of life that he encountered. So of the estimated 2 or 3 million different species that exist on Earth, he was able to get to about 11,000 different individual species. And with all these different groups of organisms, he needed some means of trying to make sense of how they're related to one another. So he developed this system of classification, the Linnaean classification system that we still use today. Now, the hierarchy that he had established allowed him to create these groups that became increasingly specific. And by the time he got down to individual groups, or what we consider species, he had this naming process. And this is what you're going to encounter throughout the book and anytime you work with anything regarding biology. We still use this binomial system. So in terms of how the names are constructed, you have two portions to a species name. The first is the genus and then the specific species epithet. Now, the genus is a noun, and then the species epithet is an adjective that describes that particular noun. So for our illustrious species, we have the species name Homo sapiens. Homo refers to man in the broadest sense, not sex-specific here, but Homo refers to man, and then sapien is wise. So here we are, wise humans. Our other Homo relatives, you have Homo habilis, habilis meaning handy. Homo habilis was handyman. They had used these stone tools. Homo erectus is upright man, so they were upright and tall. They are actually taller as a species than we are today. Homo neanderthalensis, they're from this Neander Valley in Europe. So we have this way of naming species that we still use today to be able to speak across languages and make sure there's no miscommunication in reference to specific species. But please recognize the way in which this species name is written. The genus is written first with a capital letter, and then the species epithet is written second with a lowercase letter. Now, ideally you can italicize the species name, but like if you handwrite something, then you would underline it. Alright, so how did he put all this together? Well, like I mentioned a moment ago, he created this hierarchy. So he had very inclusive groups, these sort of broad groups, and then looking at characteristics of subgroups, he created these increasingly specific subgroups to the point that you get this population that is unlike any other, and that's the point of a species. Now today we have a 3-domain system referencing bacteria, eukarya, and archaea. Now, back when I was in high school, we had this 5-kingdom system where bacteria were looked at as monarans, and then you had all these other eukaryotic organisms, the protists, the fungi, plants, and animals. Now, we now are, I guess, we have this domain system because we found that archaea, which had been lumped together with bacteria, are actually distinct and genetically more closely related to us, eukaryotes, than our traditional bacteria. Now, in terms of visualizing this system, You know, you have all sorts of references to nesting and whatever the case may be, but I like to think of it as stacked cups. So we start with this domain that is incredibly broad. Is it a bacterial cell, an archaean, or does it have a membrane-bound nucleus and belong to eukarya? Now, within the domains, or particularly our domain, the eukaryotic domain, we have other groups like the kingdom, so think the plant kingdom, animal kingdom. Then we have phylum, class, order, family, oh it's getting small, genus, and then species. Okay, well, glad I didn't go to art school. Let's see here. So the thing about the nesting here, the idea behind the nesting is that you have incredibly broad terms or groups like domain. So you cast a wide net. Do you have a membrane-bound nucleus? A, join us. And then do you have to eat other organisms for food? Okay, well, you're going to be a member of the animal kingdom. Do you have a dorsal hollow notochord, even as an embryo? Okay, well then you're going to be in the phylum Chordata, and then you get to these increasingly exclusive groups until you get to, again, this point of species, the most exclusive group of all. Okay, so think inclusive and then exclusive in terms of this hierarchy that has been established. All right. Now, in terms of how we look at life, this is a nice diagram because it sort of lays out the relationships that exist. A lot of times life has been referenced as a tree. In Darwin's book, The Origin of Species, he looked at this tree of life, this rooted tree of life. And genetically, in reality, there's probably been a slightly different situation than this one original organism that gave rise to all these different branches. In reality, we have these different domains, but there has, in history, been some gene transfer, this horizontal gene transfer or lateral gene transfer that occurred between the different domains. We know that by looking at our own cells. We have mitochondria and chloroplasts found in the domain eukarya that are derivatives of bacterial cells that had invaded one another billions of years ago. So there's gene transfer between the bacteria domain and the eukarya domain. And there's also been gene transfer between archaeans and bacteria as well. So it's better maybe to think of life as this ring giving rise to these different prongs. Now, in terms of what happens, okay, there's gene transfer, but obviously if you're going to create these different domains, we need to have reproductive isolation. So while there was gene transfer early on, obviously reproductive isolation occurred, and we wind up with these vastly different groups, particularly with eukarya. All right, so here's how we look at life. We have the little single-celled organisms up here without a nucleus, the bacteria and archaea, and then we get all this loveliness here in eukarya. All right, so this is what we're doing present day. It used to be that most classification was done by morphology. You look at the organisms. These look like they're more closely related to one another. Oh, these have 4 limbs and a vertebral column. So a lot of the early classification was just based on morphology. So initially all life was lumped into 2 big categories, 2 kingdoms, plant or animal. Well, then with better microscopic technique we discovered, oh wait, they're bacterius, and they had a 3-kingdom system. Again, by the time I rolled around going through high school, we had this 5-kingdom system that we were looking at, and people were debating whether or not there should be more. But now we have this 3-domain system that has been used. All right. A good example of why we need to have a system that is not strictly based on morphology is looking at organisms like these moles. You have a marsupial mole and placental mole. Again, on their face, you know, in terms of exterior anatomy, quite similar. You'd think they're very closely related to one another. But when you look at the genetics, the marsupials are not particularly closely related to the placental moles. It's just, again, an example of convergent evolution. All right, so let me shift this over here slightly. You can see a little better. So now again, we have the ability to apply our understanding of genetics to classification, and what that does is give us a much finer criterion we can use to look at relationships between groups. So what you want to do is basically establish how similar are the DNA sequences between species. Let's see. If you look at these two original sequences, we see that we have two separate species. You can consider these separate but related species that have homologous stretches of DNA. Now, if there's no gene transfer between the groups, then over time you're going to have mutations occur. So you may have a base deleted, or you may have an insertion of a codon, and over time that's going to lead to diversity in the sequences of the two species. But using all sorts of computer programs, you can basically adjust for mutations that occur and identify homologies between sequences. So it allows us to, there are computer programs that can allow people to look at sequences of vastly different species and identify where there are similarities. So it gives us an ability to look at relationships. All right, now this is interesting because what that in essence does is give us the opportunity to look at changes that have occurred over time between different groups to give us a sense of how long groups have been separated from one another. So you can literally build what are called molecular clocks simply looking at rates of mutation within groups of organisms. Now, a few important points to make with this. One is everything's an estimation. Okay, we can look at rates of mutation that have existed historically and extrapolate back in time, when the particular sequence or species may have arisen based on these number of mutations. Now, particular genes within a species may mutate at different rates. If looking at a gene common to different species, it may mutate at different rates. So, there are definite qualifications that need to be made with this. So what they try to do is look at multiple genes. So rather than just looking at one particular gene, you look at multiple genes and get a sense of rates of change, and then you get this sort of best fit curve that gives you an approximation. And if you can couple with that fossil evidence, then you have a fairly tight fit, or you can establish a fairly tight fit and be confident in your predictions. Now, another point I wish to make is the particular genes you look at are important because some genes mutate at very slow rates. All life uses ribosomes. So ribosomal RNA obviously is very important to the existence of life. So rates of mutation for ribosomal RNA, or genes that code for ribosomal RNA, I should say, occur at relatively low rates because any sort of mutation is probably going to be lethal for the organism, and so over time the rate of mutation and evolution is relatively slow. So if you want to look in deep time, far back in the history of life, then looking at genes for ribosomal RNA or rRNA is quite helpful. If you want to get a sense of how life has diverged in more recent evolutionary time, then you look at something like mitochondrial DNA. We have mitochondrial DNA that mutates at a relatively high rate. So if you're looking at something over the past 500,000 years, then that would be a more appropriate gene to look at. So since different genes mutate at different rates, it gives us sort of different opportunities to look back at the changes in life that have occurred. All right, we'll pause at this point and then look at how some of these genes change.